Good day everyone, my name is Lovely Marie Lopez and today we're going to be talking about the aspect of gene therapy. Medical science has detected many human diseases related to defective genes. So, when we say defective genes, it is the errors in our genes that control cell division that can cause cancer. These type of diseases are not curable by traditional methods like taking readily available medicines. Gene therapy is a potential method to either treat or cure genetic-related human illnesses. We all know that cancer cannot be cured by taking available medicines. That's why the gene therapy is one of the potential methods to treat or cure a genetic immune illnesses, like the cancer. So, in 2015, a team of researchers at the Harvard Medical School and the Boston Children's Hospital stated that they were restored a higher level of hearing down to 25 decibels, which is actually equivalent to a whisper. They used an improved gene therapy vector developed at the Massachusetts Eye and Ear that was identified as ANC80, which enables the transfer of genes to the inaccessible outer hair cells when introduced into the cochlea. So this gene therapy is usually for the deaf. Human gene therapy was actually first realized in 1971 when the first recombinant DNA experiments were planned. It can be simply viewed as insertion foreign DNA into a patient tissue that hope to successfully eradicate the targeted diseases. It was actually inspired by the success of recombinant DNA technology, which occurred over the last 20 years. Without a doubt, gene therapy is the most promising yet possibly unfavorable medical field being studied. A gene therapy has several approaches, and these are replacement of mutated gene that causes disease with a healthy copy of the gene, inactivation of a mutated gene that is functioning improperly, and introducing a new gene into the body to help fight a disease. So, if a mutated gene causes a necessary protein to be faulty or missing, gene therapy may be able to introduce a new copy of the gene to restore the function of the protein. So, a gene that is inserted directly into a cell usually does not function. Instead, a gene is inserted into another gene using a carrier or a vector. At present, the most common type of vector are viruses. The viruses are modified so they can cause disease when used in people. Some types of viruses, such as retrovirus, integrate their genetic material into a chromosomes. In human cells, other viruses such as adenoviruses introduce their DNA into the nucleus of the cell, but DNA is not integrated into a chromosomes. Although a gene therapy is a promising treatment option for a number of diseases, the technique remains risky and is still under the study to make sure that it will be safe and effective. Gene therapy is currently being tested only for the diseases that have no other cures. The stem cell gene therapy and somatic stem cells will be discussed by Bless Salan Morin.
Good day everyone, I am Blessed Morin and my assigned topics are stem cells, gene therapy, and somatic stem cell. Stem cells gene therapy. So let us tackle first what are stem cells. So stem cells have the remarkable potential to develop into many different cell types in the body during early life and growth. Stem cells also provide new cells for the body as it grows and replace specialized cells that are damaged or lost. They have two unique properties that enable them to do this. First, they can divide over and over again to produce new cells, and as they divide, they can change into the other types of cells that make up the body. So the two types of stem cells are embryonic stem cells and adult stem cells. Embryonic stem cells are derived from the undifferentiated inner mass cells of a human embryo. They are pluripotent, meaning they are able to grow or differentiate into all derivatives of the three primary germ layers, which are ectoderm, endoderm, and mesoderm. So the second one is adult stem cells. Adult stem cells are undifferentiated cells found throughout the body that divide to replenish dying cells and regenerate damaged tissues. It can be isolated from a tissue sample obtained from an adult. So stem cells gene therapy is the use of adult stem cell or embryonic stem cell. Here are also some applications of stem cell gene therapy. First, the hemophilia treatment. Hemophilia treatment is usually an inherited bleeding disorder in which the blood does not clot properly. Second, repairing mutations. Third, heart disease treatment. Fourth, sickle cell disease treatment. Sickle cell disease treatment is a group of disorders that affects hemoglobin, the molecule in red blood cells that delivers oxygen to cells throughout the body. And the last one is the skin disease treatment. Advantages of stem cell gene therapy. It discovers treatments and cures to diseases like cancer, spinal cord injuries, diabetes, and many more. It also provides medical benefits in the fields of therapeutic cloning and also provide a healthy life. Here are the advantages of stem cell gene therapy. First, additional biological complexity brought into systems by living cells. Sometimes it also leads to mortality of individual and modify the activity of neighboring genes. So our second topic is somatic stem cell. Somatic stem cell is used to refer to adult stem cells. The term somatic refers to non-productive cells in the body. It's either in eggs or sperm. So the somatic stem cell is also called as the adult stem cell, which is one of the two types of stem cells. So somatic stem cells or adult stem cells, sometimes called adult stem cells, are found in organs or tissues, can self-renew and yield the differentiated cell types comprising that organ or tissue or multipotency and are important for maintenance and repair of the organ or tissue. So the function of somatic stem cell are responsible for replacing damaged and dead cells in the body.
Good day everyone, especially to our instructor, Ma'am Marian May Injog. My name is Hijul Anura Lopez. So my assigned topic is the two types of gene therapy. There are two types of gene therapy, which is the somatic gene therapy and the germline gene therapy. What is somatic gene therapy? A somatic gene therapy can be defined as the ability to introduce genetic material or the RNA into an appropriate cell type or tissue in vivo in such a way that it alters the cell's pattern of gene expression and to produce the therapeutic effect. Meaning, somatic cell gene therapy involves the placement of a human gene into a living person's somatic cells. Cells that do not produce the eggs and sperms that in turn produce the next generation. Somatic gene therapy would aim to cure a disease only in the patient, not in the patient's descendants. Somatic gene therapy has progressed swiftly and presently being assessed in frequent clinical trials. This therapy currently focuses on recovering that are restricted to one's tissue, including the CF and adenosine deaminase or the ADA. In near future, somatic therapy may be tried on multigenic diseases. A somatic cell gene therapy transfer or express human genes into a patient target cells, except the cells that do not give rise to the eggs and sperm. This technique aims to treat a disorder only in the person's disease, not in their descendants. Somatic transfer is carried out by three methods, such as vivo, in situ, and ex vivo. In vivo is the insertion of functioning genes directly to the human body through the bloodstream. And to cure genetic disorder is ex vivo. This ex vivo defective cell will be removed from a patient, and then they are redesigned and replacement into the patient to recover its specific function. And an in situ method is a specific organ for gene transfer will be targeted, for example, the eye. So for the second type of gene therapy, which is the germline gene therapy, okay, what do you mean by germline gene therapy? A germline gene therapy is when DNA is transferred into the cell that produce reproductive cells, eggs or sperm. This type of therapy allows for the correction of disease-causing gene variants that are certain to be passed down from generation to generation. A germline therapy is one of the gene therapy where genes are modified to cure genetic diseases. The idea behind gene therapy is to replace faulty genes with a properly functioning copy. However, this is more difficult than its sound. DNA can't just be injected into a cell. The cell would destroy it, thinking it is not supposed to be there. So, the foreign DNA needs to be integrated properly into a cell by using a vector, a carrier for that DNA. Viruses are good vectors because they are designed to invade cells and introduce their DNA. Viruses in gene therapy are engineered not to cause disease, but rather to introduce the foreign DNA to the target cells. So meaning, somatic gene therapy transfer of a section of DNA to any cells of the body that doesn't produce sperm or eggs. Effects of gene therapy will not pass onto the patient's children, while the germline gene therapy transfer of a section of DNA to cells that produce eggs or sperm. So that's all. Thank you.
Hello everyone, my name is Freno Amanuari M. Sutil from the SIT2 Block 1. So my topic is the bioethics of gene therapy. So what is gene therapy? Human gene therapy seeks to modify or manipulate the expression of gene or to alter the biological properties of living cells for therapeutic use. Gene therapy is also a technique that modifies a person's genes to treat or cure disease. Gene therapy products are being studied to treat disease including cancer, genetic diseases, and infectious diseases. So, what are the types of gene therapy products? First is plasmid DNA. A plasmid is a small extrachromosomal DNA molecules with a cell that is physically separated from chromosomal DNA and can replicate independently. They are most commonly found as small circular double-stranded DNA molecules in bacteria. However, plasmids are sometimes present in archaea and eukaryotic organism. Circularly, DNA molecules can be genetically engineered to carry therapeutic genes into human cells. The second is viral vectors. Viruses have a natural ability to deliver genetic materials into cells, and therefore, some gene therapy products are derived from viruses. Once viruses have been modified to remove their ability to cause infectious disease. This modified virus can be used as a vector or vehicle to carry therapeutic genes into human cells. The third is bacterial vectors. Bacteria can be modified to prevent them from causing infectious disease, and then used as vectors or vehicle to carry therapeutic genes into human cell tissue. The fourth is human gene editing technology. Gene editing, the ability to make a highly specific changes in the DNA sequence of a living organism, essentially customizing its genetic makeup. Gene editing is performed using enzymes and particularly nucleases that have been engineered to target a specific DNA sequence where they introduce cuts into the DNA strands, enabling the removal of existing DNA and the insertion of replacement DNA. The goal of gene editing are to disrupt the harmful gene or to repair mutated gene. The fifth is patient-derived cellular gene therapy product. Cells are removed from patient genetically modified, often using a viral vector, and then returned to the patient. And that's all for my report for the day.
Good day everyone, I am Anthony Sungsino and now I'll discuss my assigned topic, which is challenges or issue in gene therapy and how to address this issue. First of all, what is gene therapy? Gene therapy involves alternating the genes inside your body cells in an effort to treat or stop disease. Gene therapy has some potential risks. A gene can't easily be inserted directly into your cells. Rather, it usually has to be delivered using a carrier called a vector. The most common gene therapy vectors are viruses because they can recognize certain cells and carry genetic materials into the cell's genes. Researchers remove the original disease-causing genes from the viruses, replacing them with the genes needed to stop disease. This technique presents the following risks. First, unwanted immune system reaction. Your body's immune system may see the newly introduced viruses as intruders and attack them. This may cause inflammation and severe cases or organ failure. Second, targeting the wrong cells. Because viruses can affect more than one type of cells, it's possible that altered viruses may affect additional cells, not just the targeted cells containing mutated genes. If this happens, healthy cells may be damaged, causing other illness or disease such as cancer. Third, infection caused by the virus. It's possible that once introduced into the body, the viruses may recover their original ability to cause disease. And fourth, possibility of causing a tumor. If the new genes get inserted in the wrong spot near DNA, there is a chance that insertion might lead to tumor formation. Other challenges faced in the field of gene therapy include: most gene therapies are short-lived, meaning patients need to undergo multiple treatments. Therapeutic DNA needs to be function in the long term and cells containing it need to be long-lived and stable if the therapy is going to provide a permanent cure. And there are two types of gene therapy, the somatic cell gene therapy and the germline gene therapy. And here is some issues. In somatic cell gene therapy, first, getting genes into target cells is tricky. Second, treatment is short-lived, has to be repeated. Third, difficult to get gene functioning in the genome. And fourth, only the actual patient affected. And in the germline gene therapy issues, first, more straightforward to deliver gene into cell. Second, all subsequent cells will have functioning gene. Third, considered unethical. Fourth, genetic manipulation passed in the children will be good or bad thing.
Climate change is the bad news story that isn't going away. From heat waves to hurricanes, from droughts to floods, extreme weather events are becoming more frequent, putting lives at risk. Human activity is causing our climate to change. The CO2 emissions from our cars, our industries, and our power plants are heating up the planet. To stop the planet getting warmer, we need to reduce the use of fossil fuels and increase the share of renewable energy, like from wind, solar, and low-carbon sources. Climate action is everyone's responsibility.
And I see this all again, from the start until the end, and I've waited for so long.
What is climate change? Climate change is a change in the usual weather found in a place. This could be a change in how much rain a place usually gets in a year, or it could be a change in a place's usual temperature for a month or season. Climate change is also a change in Earth's climate. This could be a change in Earth's usual temperature, or it could be a change in where rain and snow usually fall on Earth. Weather can change in just a few hours. Climate takes hundreds or even millions of years to change. Climate change does matter because it affects food production, global production supply chains, extreme weather events, water supply, and many other elements of the complex network of resources and institutions that make our lifestyle possible. We know many of the steps we need to take to mitigate climate change. But the impact of those steps on economic and political life are less understood. We need to make the transition to a renewable resource-based economy in a way that facilitates rather than impedes economic growth. Any other method could result in political instability with unpredictable outcomes. The transition will be long and difficult.
Good evening to each and everyone, especially to our instructor. I am Christian Sindrias Remolio from Remedios Danao Bohol, BSIT2 Block 1, and this is my evaluation about the causes and effects of climate change. Cause of climate change. The American Association for the Advancement of Science has stated that scientific evidence is clear. Global climate change caused by human activities is occurring now, and it is growing threat to society. These are the few of the ways humans are accelerating climate change. Number 1. Greenhouse gases. Greenhouse gases play a vital role in the Earth's climate cycle as the planet gets heat with the sun's rays. Some of the energy is absorbed and the rest of the energy and heat gets reflected into space. Greenhouse gases in the atmosphere trap the reflected energy, redirecting it back down to the Earth and eventually contributing to global warming. Number 2. Solar activity. Solar activity as does play a role in the Earth's climate. while the sun does go through natural cycles, increasing and decreasing the amount of energy that it emits to the earth. It's unlikely that solar activity is a major contribution to global warming or climate change. Number 3. Agriculture. There are many significant ways in which agriculture impacts climate change, from deforestation in places like the Amazon to the transportation and livestock that it takes to support agricultural efforts around the world. Number 4. Deforestation. Deforestation and climate change often go hand in hand. Not only does climate change increase deforestation by way of wildfires and other extreme weather, but deforestation is also a major contribution to global warming. According to the Earth Day Network, deforestation is the second leading contributor to global greenhouse gases. And these are the effects of climate change. From melting glaciers to more extreme weather patterns, people everywhere are beginning to take notice of the real impacts of climate change. While some nations around the world are taking action with initiatives such as the Paris Climate Agreement, others are continuing business as usual, pumping millions of tons of carbon into the atmosphere year after year. While the long-term consequences are still to be seen, for now, climate change continues to cause extreme weather as well as safety and economic challenge on a global scale. These are the few effects of climate change. Number 1. Extreme weather. Change to weather are perhaps the most noticeable effect of climate change for the average person. Number 2. Health. There are many ways in which climate change could impact people's health, depending on age, location, and economic status. Number 3. Negative impact in ecosystems. Ecosystems are interconnected webs of living organisms that help support all kinds of plant and biological life. Number 4. Water and food resources. Climate change could have a significant impact on food and water supplies. And lastly, number 5. Sea levels rising. Rising sea levels could have far-reaching effects on coastal cities and habitats. Increasing ocean temperatures and melting ice sheets have steadily contributed to the rise of the sea levels on a global scale. And that's all. Thank you.
Climate change is happening now, and it's the most dangerous and serious threat to life on our planet. Fortunately, those are the plan of solution to climate change. They are well understood. In 2015, world leadership signed a major treaty called the Paris Agreement to put those solutions into practice.
Guys, inong Gabi daw, may bitin-bitin sa ka.
For to all climate change solution is reducing greenhouse gas and emission, which the most get to zero as soon as possible, because both forests and ocean play vital important roles in regulating our climate, increasing the natural ability of forests and ocean to absorb carbon dioxide can also help stop global warming. The main way of stop climate change are to pressure the government and business to and these are the solution for that keep fossil fuels in the ground, invest the renewable energy, which to substance transport, help keep help us keep our home coast, improve farming and restore nature to absorb more carbon, protect forests like the Amazon, protect the ocean, reduce how much people consume, reduce plastic. And those are the main way of reduce of to stop climate change. Thank you.
Good day, everyone, especially to you, ma'am. By the way, I am Janine Solmiano Tolid. Today I will tackle about the climate change. So, do you have any idea about what is climate change? So, it is the global phenomenon of climate transformation characterized by the changes in usual climate of the planet regarding to the temperature, the precipitation, and the wind that are especially caused by human activities or our actions. There are many natural, anthropogenic, human-induced factors that contribute to climate change. Climate change has always happened on Earth, which is clearly seen in the geological record. It is rapid rate and magnitude of climate change occurring now that is of the great concern worldwide. So here we are the sample of climate change, which is the typhoon. So the typhoon is the principal mechanism by which climate change generates stronger. Typhoon is the higher temperature of the world oceans, or the water vapor or increasing so the effect of typhoon is the range from the structural damage to the trees, watercraft, and buildings, or both immediate and long-term impacts of human life and livelihood. So typhoon also killed thousands of people, and that's all.